Great. Hi, everyone. Welcome to the Seminar Series 5, Spatial Omics Seminar Series 5 again. And today we are very excited to have Professor Denis Wirtz and Ashley Kiemen from Johns Hopkins University. And very quick biography, Professor Denis Wirtz is currently a Theophilus Helly Smoot Professor in the Department of Chemical and Biomedical Engineering and Vice Provost for Research at Johns Hopkins University. And he is known for his studies of the molecular and biophysical mechanisms of cell motility and adhesion and nuclear dynamics in health and disease, and with a special focus on aging, cancer, and progeria. And Dr. Wirtz directs the Johns Hopkins Physical Sciences Oncology Center and co-directs the Cancer Neurotechnology Training Center and many other NIH-funded institutes. And also we have Ashley. She is a PhD candidate in chemical engineering at Johns Hopkins, and she calls herself as amateur 3D tissue reconstruction special effects. And without further ado, I'd like to turn to the Dr. Wirtz and Ashley, please.
Thank you so much, Ahmet, and thanks so much, Rong, for inviting us. And indeed, it's a little bit of a different format, not only because we're doing this remotely, but also I think there's value in having the person who's done the work to actually present the work. And so I thought it was only fair to have Ashley come along. And so thank you for agreeing to have her as well there. And so the motivation in our lab, Ashley, you can go ahead. Yeah, the motivation in our lab has been long-standing in that we think there's a lot to be learned from assessing the form of object, the shape of object, the size, the volume, the morphology. And it's not a new idea, of course. My inspiration has been for a long time Andreas Vesalius, a fellow Belgian, when Belgium was not Belgium, who at night, at the time where this was more than frowned upon, it was dangerous to take these corpses of these poor people who had been hanged in public square and really examine different systems, including the lymphatic system, the blood vessel system. And it's truly remarkable how much he was able to deduce from these morphological shape assessments and really the functions. Another inspiration, but much later, is of course the famous Form and Function book of Darcy Wentworth Thompson. We've demonstrated in the recent past of how the shape of cells can tell you a lot about the metastatic potential, the motility, the senescence status, etc. But we wanted to zoom out and start looking now at much larger structures, not one cell at a time, but really eventually what is going to be centimeter cube of volume of tissue. We used pancreatic cancer as a model system, but really what we're going to present today is amenable to any type of tissues. There's a lot of versatility associated with this technique. If you type on Google, you should do the test. You type skin 3D, right? Can we look at what has been done in assessing the kind of microanatomy of the skin, the eye, the pancreas in 3D at single cell resolution? So going from the multi-centimeter scale down to micron, even submicron, it's all cartoons, right? It's really remarkable how much we've, collectively, pathologists, histologists, and many others have deduced from really essentially 2D sections of eminently 3D objects, right? And it was time, I think, to subject a field that has, been long gone, anatomy, right? Or the Department of Cell Biology and Anatomy, they had to drop the word anatomy a long time, but really to revisit anatomy, subjecting it to modern tool of single cell sequencing, AI, and you'll see how we're learning something new here. As I said, our model system is the PDAC, pancreatic tumor, in part because we have a wonderful team. I can line up a dream team around me and my students people like Ralph Ruben, really world summit in PDAC pathology, Laura Wood, world famous work on the genetic genesis of PDAC and others. So it remains a very unmet need, where essentially the survival of patient hasn't changed in decades. It's still basically hovering about 10% for 5-year survival, and the majority of that is actually associated with cancer metastasis. All right, so that's the excuse. So what's the state of the art? What's standard is basically to take a section or two, and out of these sections stained with hematoxylin eosin, to completely standard dyes, extract as much information as way to at least determine the general composition of this section, or better, to establish a diagnostic or even potentially a prognostication of the disease. So if you have cartoon on the right, what can be seen on the left? You see cross-sections of ducts, cross-sections of blood vessels, of acini, a little bit of extracellular matrix in between. But of course, if you were to tilt this section, you have no sense, before you do that, if these different cross-sections actually are connected with one another. No sense is the pink section on the left is actually eventually connected to the pink section on the right. What that matters, if you're especially interested in metastatic disease and the transfer of cells from this primary tumor, eventually for pancreatic cancer to the liver, if those cells will have maybe a similar or common genetic background. If they're not morphologically connected, most likely they won't. Well, so can we improve from those 2D assessments in terms of assessment of connectivity, morphology, spatial proximity? In other words, immune cell infiltration. All of those, right, are typically done in 2D, which of course, even on the face of it, makes no sense, right? You can't assess connectivity. I think I've made the case, but we'll show you how assessment routinely done in clinic of immune cell infiltration, macrophage, T cells, leukocytes, really should never be done, because the next slide over a bit on the top of it or a little bit on the bottom of it, tens of microns away of that one section you happen to be examining, could be a completely different level of inflammation, level of proximity of these different structures. So hopefully these 3D assessment, which are really at the infancy at this point, would lead to improved diagnostication, improve organ on the chip. We have project where we're looking at state-of-the-art tissue engineering construct of the bone, of the liver, and we compare to the organ of origin, right? People don't ever quantify how much distant these final structures they can make on the chip really resemble the organ of the origin. We now have an ability to do so. And hopefully, for those interested in this, improve computational models of tumor progression. So what's the state of the art? Well, it's mostly clearing and light sheet microscopy, where you label already thinking about ahead of time what could be molecules of interest or cells of interest. And here we label K19 cells and E-cadherin. We're interested in E-cadherin. And indeed makes for beautiful movie. You clear first and then use light sheet microscopy. There's major limitations of this technique, even though it's non-commercial. One is that it's the millimeter scale. We'll show you we can do centimeter scale of tissue and how it matters. It's limited to typically 3 to 5 labels. For skin tissue alone, we can do tens of labels. It's not easily quantifiable. As I said, it makes for a nice movie. I wouldn't be able to tell you what to do out of those movies besides telling you there's more E-cadherin rich cells in this leading edge of these spreading tumors. The information is not transferable. Once you've done a tumor, you can't then kind of collect enough information to know, accumulate this information to go faster, to go easier, to be more accurate for the next tumor, or God forbid, for the next type of tumor. Each time you deal with a new type of tissue, a new type of organ, you have to re-optimize your clearing protocol. You have to re-optimize your light sheet microscope. And it's not not updatable. damn it, where once you've looked at a tissue, right, you have known that tissue, you store it, and you say, damn, I'd like to add a sixth label. Well, you're out of luck. You'll have to start from scratch and do a new tumor. We're going to show you data that were extracted from tumors and healthy tissues that were sectioned 10 years ago and labeled sometimes, 15 years ago. And we, after some manipulation, can re-examine and, as I said, update information that can be obtained from these sections. All right, go ahead, Ashley.
Yeah, thank you. So my name's Ashley Kiemen. I'm a student in Denis' lab, and I think I'm one of the lucky PhD students that when Denis is invited to give all these talks, he lets me come along with him. So I've had the chance to show the things I've been doing in my thesis to many people. So I'm excited to be speaking to you all today. And I'll be talking about a method that we've been developing, which is called CODA. And CODA is based mainly on tissue sectioning and H&E staining. So H&E is a common histological stain, which has been in use since about the 1920s. And I have an example H&E section on this slide. Mainly H&E has been used in diagnostics. So you could look at a section and decide if it's benign, malignant, what kind of tissue it is, and things like that. And since the, 1980s and 90s, with the advent of digitization techniques, the field of computational pathology has developed, that we can now digitally analyze these tissues and look at things like cell composition and densities, and use AI methods to detect tissue components automatically. So specifically, the application that I'll be talking about today is the human pancreas. So I like to include this slide just to show some of the specific healthy and diseased tissues that we'll be looking at. So mainly we'll be looking at pannin and PDAC, which develop inside of the ductal system. So on the bottom left on this slide, I have an H&E section of what a healthy duct looks like. And again, because this is two-dimensional, what you see is a 2D plane of the inside of a tube. So in this section, the blue dots will be the nuclei, the epithelial cells, and then the pink is the collagen that encases the duct. And then outside of that, we have some things like acini and blood vessels. Pannin is a benign neoplastic lesion which develops within these epithelial cells. When you have pannin, you have swelling of the nuclei and cytoplasm, and mucinous deposition inside the nuclei. IPMN is also a benign neoplastic lesion, but it develops within the biggest ducts. And as soon as these lesions, pannin and IPMN, invade outside of the ducts, they become PDAC, which is malignant and deadly. So next, I want to explain why we didn't simply stick with 2D analysis. And I think the slide on the left shows this well. So you can see in this 2D section that we have many ducts in this slide. And what you cannot see based on this image is, on the next slide, maybe these ducts connect, maybe two of them connect, maybe they don't connect at all. So in study of things like pannin and PDAC, which involve these ducts, it's essential that we know the connectivity of them and we know the spatial association with things like blood vessels and fat. So with that in mind, I've been developing a technique within the lab named CODA, which uses small excised pieces of tissue, and we use these to build these digital tissue models, which we can then quantify. So to begin with, again, we take small samples of tissue, we consecutively section these, and to begin with, stain them only with H&E and scan them. The next step is tissue alignment, to take independent 2D images and build them into a digital tissue volume. Following this, we've tested out a few annotation techniques to be able to label these H&E images. And finally, we use computational image analysis to add in single cell detection. So the sum total of this gives us a quantifiable digital tissue volume. And I'll quickly step down each one of these techniques and then jump into some analysis. So this slide, I hope the video is playing well on Zoom, is meant to show the qualitative quality of the image alignment that we do. So on the left, what you see is moving down the Z stack of these H&E sections if we just laid them on the slide, scanned them, and then compiled them into an image sequence. So Obviously, the tissue is jiggling and moving because when the technicians laid them on the slide, they didn't lay them in the exact same position each time. Next to this is following the elastic image alignment. So we take into account global movements of the tissue as well as small pulling and folds in the tissue so that we can have well-aligned objects like ducts and blood vessels. So at the end of this, we now have a digital volume, but it's still completely unlabeled. We can make these beautiful little H&E models, but that's it. Nothing is quantifiable yet. So the next step was involving stain deconvolution. So taking into account the bimodal staining of H&E, we can calculate the optical densities of the two stains and demix them into a hematoxylin image and an eosin image. And because hematoxylin mainly stains acidic components like DNA, it's highly intense inside of the nuclei of cells. So in this middle image, you see black spots on all of the nuclei. And by detecting the two-dimensional intensity minimums in the image, we can identify all the cells. So following this, we now have an H&E volume and we have the location of all the cells in the tissue. But we don't know the identity of the cell. So we don't know this is an epithelial cell, this is a fat cell, and so on. So next, when I was new to the lab and didn't know a lot of computational techniques yet, I began just by manually annotating these pannin, the mutated epithelial cells, which, as you can imagine, was quite time consuming. But by doing this, I was able to make these little visualizations of the pannins. You can see in this image, we can see the connectivity, we can count the lesions, and we can quantify things like volume. But if you notice how small of a zoom in this is that I'm showing you that contains these pannin, you might ask, what about the whole image? What about the blood vessels? What about the healthy ducts? And given that these annotations took me almost a full day to do on 1,000 tissue sections, it would be impossible to fully annotate this block by myself and not a well-spent PhD. So instead, we began looking into a semi-automatic technique, and I took advantage of an existing semantic segmentation model called DeepLab and adapted this to analysis of histological images. So in a sample of containing maybe 1,000 H&E sections, we would pull out 7 of those and manually annotate, say, 50 examples of each of the tissue types that we'd like to identify. Next, we would cut these out of the image and compile them into little tiles, which we could use to optimize a semantic segmentation model, which could then be used to fully annotate all of the images in the sequence. Once we have all the independent 2D images labeled, we can compile these into now a fully labeled tissue volume. And up to this point, I think I've analyzed maybe 26 independent samples. And the good thing about AI methods is that because it's based on manual annotations, each time we build a new model and gain a new sample, we add to the total database of annotations that we have. So with my newest models, I'm not doing any annotating at all. I can just collect the specimen, section it, scan it, and label it automatically. So next, I'd like to show this video just to give a sense of the things that CODA can do. So this is showing the H&E labeled block and then showing each of the components one at a time. Being shown now is blood vessels, the healthy ducts and pannin. And now it's going to zoom in on a single duct to show you connectivity in detail. In pink is collagen. This is blood vessels again, islets, which make insulin and glucagon, fat, acini, which make digestive enzymes, and then zooming in to show that by quantifying the cells and by labeling the H&E, we can identify the identity of each of the cells now. So maybe I'll just let this play again and then move on. So this is all 7 labeled components, ducts and blood vessels, ducts again, and then we'll zoom in and show some of the components. And people have asked me what's about the size of one of these tissue volumes, and we quantified it once and wanted to think of a good analogy, and we found out that it's about the size of 2.5 nickels stacked up, which I used to think was small. I've been told that's actually quite big as tissue modeling goes. So moving ahead, we've analyzed many samples, but I like to focus on just a few because each one of these I could spend a whole day talking about. They have so much data in them. But instead I'll just focus on a healthy sample, a sample containing pannin, and then 2 samples containing PDAC. So as a whole, I like to point out a couple of things. So in this table showing the cell density of each of the components in some of these samples, we saw that as pannin and PDAC develop, we see a bulk loss in acini as those cells begin to die, and new deposition of collagen. And as an effect of this, the total cell density in these tissues is going down significantly. You can see between the healthy sample and the PDAC sample all the way at the end that has no acini left, the cell density has gone down almost 7 times. So focusing in on a couple of the samples one at a time, just beginning with the healthy system, I like to show this video, which in blue is the pancreatic ductal epithelial cells. So because this is the ducts connect to these big tubes, the shapes can be kind of unexpected. So you can see this one, we have this big U-shaped duct with small ducts coming off of it. And then I show next to this what an H&E section of one of these ducts looks like. So a question that we had when we initially plotted this was, because of the kind of unexpected shape of these ducts, how is this affecting the collagen and the way we see the collagen in these 2D slides? Because we've seen in the past, many labs who do digital pathology analyses have done things like collagen alignment assays in 2D. So as an example, I want you to think about 2 ways we could cut this duct. One is axially sectioning this duct, and the second is longitudinally sectioning the duct. So in 5 of these samples, we went in and we identified locations that the histology was cut axially and longitudinally, like so. And then in each of these locations that we identified, we again unmixed the H&E images. And by looking at the eosin channel, we could quantify the collagen alignment. And what we found was that the collagen was typically not well aligned in the axially sectioned ducts, which you can even see just looking at the sample histology on this slide, and that it was well aligned in the longitudinally sectioned ducts, both the collagen and the elongation of nuclei that we identified. So what this is saying is that if we go back to this image of the ductal connectivity, and if you look at this big U-shaped duct in the middle, what we believe is that the collagen aligns itself to the angle of the duct, and that depending on how the duct is sectioned, you may see that alignment and you may not. And the big point of this is that a lot of these quantifications that teams do on 2D histological images often don't take this into account. So people don't think about how is the angle of this object, which you may not even know what that object looks like if you only have one slide, how is it affecting all these calculations that we do? So that's one thing that CODA has definitely been able to help with.
They're simply completely erroneous on the face of it. Ashley is more nice than me, but they're simply wrong. Any second harmonic generation or simply labeling of 2D sections to assess collagen alignment is going to be wrong.
So next, I want to show a little bit about one of the samples that we analyzed, which contains pannin. And just to go back quickly to these definitions, pannin is a mucinous neoplasm that develops within the ductal system and has been known to extend out of the ducts and into the acini, but mainly develops in the ducts. So in this video, I want to exemplify something that we've done in analysis of pannin, which is to not only label all of these lesions, but to identify spatially distinct pannin. So in this one, in blue is the healthy ductal epithelium, and then in pink is the biggest pannin that we see. In cyan, light blue is the second biggest, and so on. So you can see in this that in these small-ish tissue volumes, many pannin can develop. And in this one, I only label the 10 biggest, but we found 57 in this model. So in subsequent analysis of many samples, something that we've done, because looking at these big tissue volumes can be kind of a lot sometimes, is we'll sum the image along the Z dimension to take us back to 2D. because at times it seemed that going back to 2 dimensions, like this image I'm showing now, helps show big changes to the tissue. So what's going on at kind of a global scale. And what we saw in analysis of many samples containing pannin, about 22 of them, is that we could divide them into a few phenotypes. So this one that I just showed, we have mainly tube-like pannin that seem to copy the layout of the existing ductal systems. You can see all these pannin just look like thin hollow tubes, just like the blue lines do. A second phenotype that I'm seeing is these clumpy pannin that seem to extend out of the ducts and begin to look like the acini. So the acini of the pancreas will exist in these small clumps like this, and they connect to the ducts almost like these clumpy pannin do. And then the final one that I'm seeing, which I just think is so beautiful, I always have to include it, I call them balloon-like pannin, and these develop within dilated ducts when you have a tube with essentially a balloon coming off of it, that it's a hollow ball. So based on all of these, what we saw was that the tube-like pannin exist within the ductal system, as do the balloon-like pannin. It's within a dilated duct, and the clumpy pannin exist within the acini. So I've made this video to kind of exemplify this. This is a ductal pannin that the layout of the pannin cells copy the layout of the healthy ductal epithelium. And then this is a lobe-like pannin that the cells extend into the acini and take on the shape that the acini used to inhabit. And I'll just let this play a second time. So it had been known in the past that pannin can extend out of the ductal epithelium into the acini. What had not been known is that this affects the global shape of the lesions. And we've since begun to quantify how this changes things like inflammation, how it changes the size of the pannin. The biggest ones tend to be in the ducts, but also the biggest ones always contain both phenotypes. So we can do a lot of these calculations that simply could not be done in 2 dimensions. And what we hope to eventually do is to tie things like inflammation and size that we can pull out with CODA to which of the pannin would be the most likely to eventually invade into the tissue and become PDAC. So next, I'd like to talk about a sample that does contain PDAC and some analysis that we've done with this. So in this video, I'm just showing you the location of the PDAC cells in this sample. It's the yellow down at the base, and again, the blue is healthy ductal epithelium. And in this sample, what we saw is that the PDAC was mainly existing within the ductal system, and then we have these small clumps, these bunches of cells kind of pushing off of the main mass. So we wanted to look at these specifically and ask, what is it about this location in the tissue that's allowing these cells to invade distally instead of just slowly expanding like the bulk mass is? And what we found at the biggest of these locations was that the cells seem to hug along the outside of a duct. So again, the blue is the healthy duct in this. But when we looked at the histology, which is one of the biggest benefits of CODA, that we can always go back to the H&E and show it to a pathologist, it does not actually invade back into the duct. So if you look at this image, this outline in the middle is healthy ductal epithelial cells. And then we have all this PDAC sitting outside of the duct. And pathologists looked at this and told us that they expected within a couple of sections that this would invade into the duct, because that's what they always expected. And instead we found it doesn't, and it's just moving along the outside within the collagen. So I was looking online to see what people have said about this in the past, and I found that in patients with highly aligned collagen, they tended to have bad outcomes. So patients with aligned collagen wouldn't live as long. And it's also been hypothesized that PDAC can move easily along these highly aligned collagen locations. But on the flip side of that, I found in a study of healthy pancreatic ducts that people believe these ducts do not have well-aligned collagen. But if we go back to what I told you a few slides ago, that we believe people who have done these 2D collagen alignment studies don't take into account the dimensionality of the ducts and the angle of sectioning. We believe that these ducts do have highly aligned collagen, which is allowing this PDAC to move along the outside of the ductal system instead of only being able to invade inside of the epithelium itself. And having found this phenomenon in one sample, we've since found it in all of the PDAC samples that we analyze with a little bit of digging. Finally, I'd like to talk about the last sample, which also contains PDAC and has a phenomena that's been studied quite a bit, I believe, but maybe not at this magnitude, which is venous invasion. So in this sample, we found a vein which begins at the bottom of the section and extends all the way to the top and is fully invaded by PDAC the whole time. So we don't know the full extent of this invasion, but we know that it's quite long and that the PDAC cells split with the vein and continue to move along it in kind of an unexpected way. I think when most of us think about metastasis, we think about cells invading into the lumen of a duct and kind of floating along in the blood and then leaving at some distant location. But what's seen often with PDAC, because it begins at these big ductal epithelial cells, is as soon as they get inside of a blood vessel, they think it's a duct again, and they begin to look like ductal cells and take on a ductal phenotype again. But something kind of unexpected that we found in this case, if you look at the plot on the bottom of this slide, was that the cell density of the PDAC within the blood vessel itself was 6 times the bulk density. So somehow these cells, once they get inside the blood, don't seem to mind being packed so tightly. Next, I'm going to hand it off to Denny to finish up.
Just as a note, not only, at least it changes my kind of cartoon version of metastasis, right? The cell sticking its neck out into the blood vessel, being sheared off. We've seen all those cartoons, I've drawn them myself. Well, in this case, it's a plug of cells completely occupying these blood vessels, moving like a worm into all the way it's going to be to the liver. Also EMTs, right? Well, we've read the textbooks. Well, these are E-cadherin-rich cells. That's why they're so compact. So a lot of pathologists question EMTs because they've seen such structures. What I'd like to highlight is, thanks to the 3D reconstruction, we can determine the point of entry, the point of exit, intravasation, extravasation, right? If you take random section of a tumor, one section at a time, right? It's basically, you'd have to be the luckiest person on earth to really find by serendipity that point of entry, and maybe then examine what's in the vicinity, what's in the neighborhood. Are they more or less immune cells, more or less cancerous fibroblasts, or other type of cells? And so, motivated by these questions, right, we wanted to add to our arsenal, to not just stick to H&Es. There's many, of course, types of cells that cannot be detected in H&E by an expert eye examination, right? We can take advantage of the fact that we can skip at least 2 slides. We've done a quantitative assessment, how much ultra-structural information is lost when you skip 1 slide. So H&E, skip H&E, H&E versus 2 slides, H&E, skip, H&E, skip, or 3, etc. And at 2, we're comfortable to say we preserve all the information about ducts, fat, blood vessels. But then we can exploit these intervening slides to stain them with additional marks, including immune-associated marks like CD45, CD3, FOXP3, or any other cells. Or better, and we're working with this with Rong, to know, go crazy. Spatial omics, transcriptomics, spatial proteomics on regions of interest, exactly where you want to do it. Not waste your money on the whole slide fishing for something. You can use CODA as a way to identify those regions of interest. Well, let's see what can be done and learn from alternating H&Es with these IHC for CD45, CD3, and FOXP3. Go ahead, Ashley. So first you ask, what is the distribution of leukocytes as you move away from the surface of these precursor lesions called panels, right? And if you were to take a random slide, it's all over the place. It seems to be high at the surface for some, but also low for some others, right? As you move along this meandering shape of this panning, it seems to be all over the place, right? We've drawn a basal structure, but of course, and that's the next slide, if you know ensemble of which these different profiles, you seem to then see an enrichment of these immune cells at the surface of the panem, and then a relaxation towards a basal value away from it. But then, of course, you lose the spatial information that was contained in these different slides. And of course, the solution for this, to get this quantification, is to create what we call now 3D immunomaps. This is a single precursor solution. We can do this at the centimeter cube length scales, and we've color-coded here from high to low, depending on the number of leukocytes within, I think it's a 250 micron distance away from the surface of this panem. And you can see, no surprise here, it's all over the place. It goes from very cool to very hot, from very inflamed to not inflamed at all, right? From 5,000 or so leukocytes within these regions near in the vicinity of the panem to none at all. And so just demonstrating that if you had happened to cut on the left, you'd call and annotate this panem as being cold, while if I annotate on the right with a section as being hot. And it's, of course, it's neither of those two. It goes from very low to very high. And this is just then cross-section showing you that we have hot spots and you have cool spots, not only near these panem regions, but then in the bulk of these tissues. We can quantify these, go ahead, Ashley, these at scale, now for an entire tissue, and ask, where's the highest level of inflammation? Is it near islet or inside islet of Langerhans? Is it in epithelial ducts? Is it in blood vessels, smooth vessel? And long story short, you see, while there is inflammation in panem, and we just showed you an example or two, of course, maybe as you'd expect, there's a lot more immune cells in the stroma, in the collagen structure. If you remove this, nevertheless, the highest level of inflammation is indeed found in these precursor lesions called panems for the pancreas. So this was one demonstration of how we basically coupled one other mode or modality with H&E, in this case, IHC. These things are limited by cost, of course, right? But we can use this intervening slide to do IF. We can do IMC. We're starting to do this. And better with the help of Rong, looking at a technique like DBit-seq for spatial and proteomic and transcriptomics. But again, not waste your powder on regions of little interest. We can use H&Es to determine what are the region of interest. Maybe these precursor lesions or the point of entry of the inside of a blood vessel or the outside blood vessels, and then go nuts when it's called for, thanks to Rong, to really enrich information in the volume, not spot of entry of interest in these volumetric tumors and tissues. So without further ado, we'd like to thank you for having attended this talk. And really, this is a collaboration with my partner in crime for the longest time, Patient Wu. Ashley is co-advised by the two of us. And really, as I mentioned, really the beauty of being in Hopkins, and now I'm going to be parochial, I'm a VP for research after all, but when in some times of cancers, I can line up a true dream team, and Laura Wood and Ralph Huber not only has brought tons of insight and new questions, but really, and sometimes it matters, cheerleading our efforts to really push the envelope technologically. So hopefully you've learned something. We'd love to hear your questions. I've answered some of those questions along the lines, but please feel free to ask some more.
Thank you, Denis, for this fantastic talk. Now we are ready for the quick questions and answers. And if you have some questions, please feel free to unmute yourself and ask. Otherwise, we can also help read the chat. And there's, I think, one question from Justin He. Shall we read it for you, Denis, or?
I can read it.
Okay.
Give me something. May I ask what is criteria here used to judge a cell is a cancer cell, normal cells, or precancerous cells, right? So here the ground truth is what the pathologist says it is. So it is really an expert's determination. Turns out by now we can annotate those things ourselves. There is no question what makes a cancer cells in an H&E. What is a lot more difficult is, for instance, to start delineating an immune cell from a fibroblast. We can still do that, but subtype of immune cells, forget it. That's why we had to move to IHC or other modalities. But there's no problem determining a panim from a precancerous to then a cancerous cells. There's no doubt about it there.
But later, once you integrate multimodality data, and then you can retrain your AI, right? Retrain your machine learning.
Totally. And so I have an agenda here. I want to, drive all antibody companies out of business, right? If we, at least antibodies, not sequencing yet, but if we can, I think, as I said, going back to form and function, so much information is truly embedded in these H&Es, in the morphology of those cells. I have no doubt in my mind we can differentiate those subtypes of immune cells in H&E. Actually, we've done it already. We can delineate B cells from T cells in H&E. I took a bet with Ralph Rubin that we could do it. He said, no way, Jose. Well, I was right. It's black boxes, right? Let's agree on this. I don't know what makes it really different morphologically, but those algorithms can. And a ground truth, of course, was an immunostain. Thank you, Ron.
And there's a practical question about how long does it take for a whole analysis?
So at the beginning of my PhD, it would take, I don't know, 5 months, I think the oldest one. And then that went down to 2 months, and then it went down to 1 month. And then in the past 2 weeks, I've done 15 models alone. So it's definitely speeding up. And it's getting to the point that they don't take long at all.
So Ashley, question from Sandhya Krishnan. Do you use the whole block for serial section? Of course, we have to leave it some for the pathology for clinical use, but yeah, we get a big chunk, right, Ashley?
And yes, yeah, we get a big chunk, but something we've done like with the IHC study is to hold out 2 sections. So we'll take a section of H&E, then hold back 2. And something that was not shown in the slides was that all of the samples that I've analyzed got collected at Johns Hopkins in 2010. And most of them got sectioned and stained in 2010. And we've only just in 2019 went back and added the IHC to these decade-old samples once we'd finished the H&E model. So the beautiful thing was that we could do all the modeling and then decide which additional antibody labels to add.
Oh, great question from Pankaj Moga. I love talks like this where there's great feedback. You learn even though you think you've thought about those data a lot. But do you see any preference for those cancer cells to follow small as opposed to bigger blood vessels? Ashley, have you assessed this?
So we have a second study that's focused mainly on venous invasion, and we most commonly see them invading into veins and lymphatic vessels. And maybe that's because they tend to have less smooth muscle, and the vessel is mainly made of endothelium. So it may be less difficult to invade.
I'll take Moashash questions about, have you tried other stains besides H&E? Well, of course, I showed IHC. We can do immunofluorescence as well. But in terms of 3D resolution, no, it doesn't. It helps adding, in the type and number of type of cells we can identify, of course. But of course, eventually, our ulterior motive is to get away from those more expensive modalities and go back to cheap and standard H&Es. I always say the H&E done in Timbuktu or New Haven or Baltimore, God forbid Paris, is all the same everywhere. That's what makes, I think, this technique, amenable to many other labs in the future, is that we can train algorithm to recognize in H&E cells that otherwise can't be recognized by experts.
And as we are waiting, maybe I'll also ask a quick question. So this actually reminds me the TEM array tomography, where the individual slices are put together. So there are two relevant questions here. One is the thickness. How does it really matter if it is like sub-micron, 2 micron, 5 micron, 10 micron? And then I guess there are trade-offs, but have you guys compared this? And that's the first question. And the second question is, there is also something called FIB-SEM that they do tomography of cells. They cut it and then image it, cut it and image it. Can we envision a device where you combine a microtom with a microscope?
All great questions. Maybe Ashley, you want to answer the first one? I can ask the response to the second.
Yeah, so we've tested out how many tissue sections we can skip with each of these, and Denny showed that in the IHC study we skipped 2 in between each typical histological slide. Computationally, we achieve good tissue alignment with up to 5 sections missing, but obviously it also depends on what tissue you want to study and how much data you lose, the connectivity of objects, of can you still connect blood vessels, do you lose the splitting of these ducts? So I like to keep it to 2 sections held out, but I think in
So it's also about the thickness of the section.
Oh, yeah, yeah.
And so these are 4 or 5 micron. If it's thicker, right? So you have less issue if that's an issue of shaving nuclei or cells in the middle or somewhere, right? But then you start losing your single cell resolution aspect. So I think that the trade-off is the standard 4 or 5 micron, which is completely standard. Where we're going is the opposite direction. We'd like to go to super thin section to do what I mentioned, which is to take successive sections. So that the classical way to train an algorithm to recognize cells in H&E you cannot otherwise detect in H&E is to serially stain, right? To do label number 1, you wash, label number 2, you wash. I don't want to do that. It creates folding, it creates problems and align. Forget that. What we take is thin sections successively, and they're so thin, right? A tenth of a micron or a little bit thicker, but barely. And we then have different sections of the same nucleus or the same cells. And now you can do H&E, IHC of label 1, IHC label 2, etc. And now we can train algorithm on the same cells without having to do the serial staining and washing. You understand?
Yeah.
Yeah.
So
so
second question. Yeah, the integration.
So we have a project, and I know if it's going to be satisfactory how we answer that question, where the idea is to measure fat content out of is it MRI, Ashley?
I think so. I'd have to double check.
Mimi's work. So where we take this MRI images, the 3D reconstruction MRI, of course, with more like I don't know, millimeter at best resolution, and then resect the tumor and serially cut and hopefully train algorithm to recognize in MRI what can be recognized at the pathological level. We have no idea if it's feasible. We trust these AI algorithms are powerful. They'll beat, again, the expert eye, but we don't know yet. That'd be great, right? Where you can start getting maybe at least multi-micron resolution or features being resolved for what is not the nominal spatial resolution of the MRI. So it's being done in microscopy. We're trying to do it in light microscopy to enhance resolution. We try to do this with different modalities. So MRI, of course, being clinically used all the time and make our technology more translatable to the clinical setting.
I see. Yeah, I was also referring to, how they're sliced, in the microtome. There's a slice sitting. If you put a microscope right behind it, and then you should be able to capture the tissue. Of course, that should be maybe H&E stain, but now people are doing virtual stains.
They are, yeah.
Without staining. So then basically you can just cut it up, move the slide to the next one, and almost like the strip of film, right? It's moving. the sliced ones is moving to the right side, and then you're taking picture of the left one, right?
Yeah.
So it would be very fast, I guess,
No,
if there's something.
I
like
agree. It's you know, socialist me trying to make technology that at least somewhat amenable, you know, and accessible to the masses, because yeah, if think of this, right? If I spend $1,000 per section and have 1,000 section, right? I need the Bank of England to support my research, right? And so this it's the balance I want to strike, and hence use H&E again, because so routine, so really cheap, and use only these more expensive modalities when you have a good question, when you've mapped out with CODA the regions of interest. Maybe I'm wrong. Maybe I should go ahead and spend money, but.
Yeah, I think I have two comments here. Also, there's one question in the chat box. I think Denis, you will love it. So I think my two comments. Number one, I do know. I think some company right now, I think working on this kind of combine the microtom and imaging technology. Hopefully, you will see something like that in the next couple of years. Second, I think one of my former colleagues used to work on kind of autofluorescent imaging of unstained tissue and the computational reconstruction of something that looks exactly like H&E. Yeah, that might also speed up the CODA process later. Yeah,
absolutely wrong. Yeah, we've seen this, and yeah, I hope in a way all your questions reflect this. Using yourself a lot of versatility in this technique. It's super versatile, unlike I think clearing and light sheet. Once you've done clear your tissue, you're done. There's nothing, and you've labeled, you know, 5, you pushed to 10, maybe you're done really. And again, no quantitation, small, and I've talked about it. Here, there's no real technical limitation. Like the volume, we say centimeter, but it could be much bigger. There's no limitation per se. Number of labels, no limitation either. We see a path forward to add always more more labels. Ashley said it, right? She's not even annotating it at all anymore. She can move to the next tumor and the next tissue and go ahead. We've even went for pancreas to the breast, or all completely unrelated tissue like the skin, and many subtype of cells like endothelial cells can be detected in the skin, even though trained on the pancreas. So a lot of potential. I think you're totally right, Ron. You had another comment. You had 2 comments. Sorry.
Yeah, I already gave 2 comments. But I do have another question for Ashley. I was very intrigued by your sort of the panning data. So within a centimeter scale, you have like 14 different pannings. That's very striking. I think about how tumors develop, right? So people may ask, okay, those 14 different kind of precursor tumors are identical genetically or not. So I think no one knows. Maybe there are 14 different tumors begin to develop at the same time. Yeah, if you can do kind of pick up those tumor and do genetic analysis and
yeah, it's funny you say that. We actually have a study that's ongoing with the Wood Lab at Hopkins. I don't know if maybe Denis told you about it. That they do just that we identify spatially distinct pannin in these samples, and then they use LCM and DNA sequencing. Because of the age of the tissue, it has to be DNA. And then they can identify mainly within the fully enclosed pannin, because any that go off the edge of the tissue, we can't know if they connect sometime in the outside space. But then they can label which ones have distinct genetic lineages and which ones have the same genetic mutations.
And there is a correlation where those pannins connect, or ductal structure connect, and the type of mutations they have. And so hence, you know, justifying the 3rd dimension for all these 2D assessments really can be misleading.
Yeah.
Yeah, cool. And I think the most we found is one of these samples had 92 pannin in it. And typically they have maybe 1 to 2 massive pannin, and then many small pannin close to the biggest ones. So we don't know, and this study may help with that, if maybe these small pannins seed off the biggest one as opposed to developing independently, like maybe the biggest ones can cause new pannin to develop. So that's an exciting question.
For the physicists in the crowd, I don't know if there's any, if you plot the probability of volume versus volume, it follows a beautiful power law with an exponent of -2. So P of E goes like the volume of the pannin to the -2. So a length scale to the -6. It's really strange power law, but it of course only shows that the probability to have a very large, particularly large pannin is very, very low. Well, that's the good news, because I can tell you all of us on this call have pannins in our pancreas. This is the most common precursor lesion of them all, among all type of course, we don't die all of pancreatic cancer, and that means that only a tiny fraction of them can grow and can have then enough probability to have the mutations. And I think these different type of pannin subtypes that we've identified to then move to the next progression step of a PDAC.
Yeah, I think prostate cancer very, very similar. You'll probably want to look at that as well. It's called a multifocal prostate tumor. So yeah.
We should look at this.
Yeah, I think a few questions in the chat box. Within a limited amount of time, maybe if you can, Ashley, you can take a look. I think one of those interesting, Danny, you'll probably want to take a look. You will love it.
Can I ask a question as well?
Yeah, sure.
So I know you already mentioned about incorporating this into with other spatial omic technologies. Do you see any? I guess do you predict or see any challenges with using probe-based approach or array approach? Some might be easier, some might be harder to kind of to use CODA for 3D reconstruction.
I think the big delineating step is what can be done in paraffin embedded versus fresh tissues. But again, the beauty, you can cut, save, and do spatial transcriptomics. We hope we can do this assessment quickly so that Rong can actually make, decide where to do his spatial transcriptomic and proteomics. We're working on the workflow. We're waiting. So it turns out PDAC in the US are pre-treated. Patients are pre-treated before they move to resection. So we need untreated PDAC, and actually in Germany they're not. So we're waiting for samples coming out of Germany or Holland, I can't remember. But we'll tell you, stay tuned, and hopefully that will work out. But I think that's the big delineating step.
Could you theoretically do CODA for reconstruction without having H&E stains? Like, would the principle still? You would obviously have to retrain your model, but
I mean, this is what Rong was alluding to.
Yeah.
Like theoretically, could you just do it with immunofluorescence or IHC stains?
Oh yes, yep.
Without any H&Es to supplement.
Yeah, totally.
Yeah, I've tested out on outside studies, just looking at single 2D slides of IF stained and IHC stained images, and in both of those, I can use the exact same computational pipeline and just use new annotations and can detect anything the human eye can detect in those tissues. We can automatically label.
That's awesome. Cool. Thank you.
Thank you.
Quick question from here that can complement the discussion that you had for the alignment. And so in super resolution microscopy, the field is using fluorescent beads to align the images, right? If you have like 3 beads, and in EM community, they use these patterned substrates, right? So could there be a solution where your slides maybe is patterned or something, where you would put the slide on?
We have, I believe, a much clever solution. I can call it clever because I didn't come up with it. But Patient and Ashley came up with an iterative process that is a lot better than using fiduciary markers. It's maybe Ashley, you can elaborate.
Yeah, one of the oldest samples that I analyzed, it had these funny objects inside the tissue image, and I was teaching myself about pathology just by Googling things, and finally I gave up and I went to Pei-Shun, who co-advises me, and asked, what is this? And he told me it was toothpicks that pathologists had physically put into the tissue a long time ago, because they thought we could use these stable, consistent objects to then line the toothpicks back up to get the tissue back into alignment. But what we found was the toothpicks actually caused the tissue to split at those locations. So I think that was the messiest sample that I had to align. But the method that we use now is just takes advantage of common big objects in the tissue. So in these systems, it's big things like ducts, blood vessels, and bulk fat in the sample. And we have a two-step alignment method that, to begin with, we just globally move the tissues back into place. So just adjusting the angle and the dislocation. And then the second step, because tissue is soft, I tell my students it's like deli ham. Like if you imagine how deli ham is cut, you could take it and pull it and fold it and do all of these things. So the second step of the code just physically adjusts these little pieces of the tissue back into place.
Cool. Then it will be more neat.
Better alignment. No, this alignment turned out to be a critical step and not a trivial one. And I think that there's some comments of this. People have had the idea of serially cutting this for a long time. That's why we had samples that had been serially cut 10 years ago. And when it came to like, they were trying to physically align these things using toothpick, using fiduciary markers, and it failed miserably, and also manual annotation. And it really is the combination of AI for automatic annotations and two of this alignment step that I think open the possibilities.
And it's still not faultless. I mean, obviously we can't unfold a piece of tissue that's been folded, but we can take the two unfolded halves of it and move those back into place. So I also want to say that the Johns Hopkins histology lab is just amazing, that I've seen the quality of the tissue sections go up significantly since we've been sending them, tens of thousands of sections a month to scan. So that also definitely helps.
Pretty neat. Are there any other questions? There's one more.
Oh, thank you, Mary. We've done it. It's shame on us. I love nuclear size and shape. I think there's tons of information in those, and we've developed actually an algorithm called Vampire to actually analyze nuclear size distribution in vitro and in tissues. And so I haven't looked if there is some outlier, what you call supermassive nuclei. We'll do this and acknowledge your idea. It's with super fast and precise algorithm to exactly do that. We go ahead and do it.
Great. Any other questions? If not, let's thank to the speakers again, Dr. Wirtz and Ashley, and I'm gonna use this clapping sign as well as the physical, both of them. And thank you guys so much. This was fantastic. And as Dr. Fan put, this is the jewel in the crown of spatial omics for us now. And we look forward to reading the more of the results and more of the discoveries soon. And before we wrap up, I'd like to also announce that next week, we have actually another speaker from Johns Hopkins again. And Rong Fan, she will be talking about statistical approaches and computational tools for analyzing spatial results, single-cell transcriptomics data. And with this, thank you again, Denis and Ashley. And please enjoy the rest of your week and have a nice weekend.
Ahmet, thanks so much again for inviting us. Rong, you're a great, good friend.
Thank you.
Yeah, thank you for having you, Danny, Ashley, and have a good weekend.
You too. Bye, everyone. Thank you for this wonderful feedback.
Thank you.